Imagine a computer that doesn't rely on electricity. Instead of wires and silicon, we could use the very molecules of life, like DNA and proteins, to compute, store data, and make decisions. This concept, called molecular fluidic computing, is a leap beyond traditional electronics. It promises to transform fields like medicine and biology. At its core, this type of computing uses the natural properties of molecules, such as their ability to interact and react, to solve problems. While traditional computers rely on circuits and electricity, molecular fluidic computers leverage chemical reactions in fluid environments to perform tasks like diagnosing diseases. In conventional computers, electricity flows through wires, enabling circuits to process information. But in molecular fluidic computers, information is carried and processed by molecules flowing through tiny channels in fluid environments. This system is particularly exciting because it opens up new possibilities for computing inside living organisms or in extreme environments where traditional electronics fail. Imagine a computer that could function inside your cells, monitoring your health at a molecular level, detecting early signs of illness, or even delivering targeted treatments without the need for external interventions. Instead of using electrical transistors to process data, molecular fluidic computers use molecules as data carriers. These molecules, such as DNA or proteins, flow through tiny channels called microfluidics. When they meet, they interact chemically, and this interaction acts like a computation. For example, in a traditional computer, an electric current flows through a transistor to switch it on or off. In a molecular fluidic computer, a chemical reaction between two molecules can perform a similar function, but the process involves no electricity. Understanding how these molecular interactions work requires a basic knowledge of chemistry, but the fundamental idea is that molecules can react in specific ways to carry out logical operations. Microfluidic channels are tiny tubes etched onto materials like glass or silicon. These channels are only a few micrometers wide, about the width of a human hair, and they direct the flow of molecules through a liquid medium. You can think of them as tiny highways for molecules. By precisely controlling how molecules move and interact in these channels, scientists can direct chemical reactions with extreme precision. These channels are crucial in controlling when and where reactions take place, making it possible to perform computations on the molecular level. For beginners, it's important to visualise these channels as miniature plumbing systems, where molecules are the cars flowing through them. In traditional computers, logic gates control how decisions are made based on inputs like electrical signals. For example, an A&D gate outputs a signal only if both input signals are present. In molecular fluidic computers, the inputs are molecules, and chemical reactions serve as logic gates. For instance, two specific molecules might trigger a reaction only when both are present, mimicking the behaviour of an AND gate. These molecular logic gates enable the system to perform computations in environments where electronics can't function, such as inside living cells. Understanding this concept is key for beginners to appreciate how molecular computers can process information just like silicon-based computers, but in a much more biologically integrated way. In traditional computers, switches control the flow of electricity, turning signals on and off to enable computations. In molecular fluidic computers, switches are molecules that control the flow of chemical signals. These switches could be proteins, enzymes, or synthetic molecules that change shape when they bind to a specific target molecule. When the switch is on, it allows the next step in a chemical reaction to happen, just like turning on a light switch allows electricity to flow through a circuit. Understanding these molecular switches is important for appreciating how biological systems could be programmed to compute in much the same way electrical systems do. In a molecular fluidic system, computations happen through chemical reactions. When two molecules meet in a solution, they may react and produce a detectable signal, like a change in colour or fluorescence. This reaction serves as the output of a computation, just like how a traditional computer produces output by turning on a pixel on your screen. Beginners can think of it this way. Each chemical reaction is like a tiny calculation. The result of the reaction, such as a molecule changing shape or emitting light, represents the answer to the problem being solved. Understanding chemical reactions in this context is essential because they are the fundamental building blocks of molecular computation. DNA is a remarkable molecule, not only because it stores all the genetic information for life, but also because it can store vast amounts of data in a very small space. 1 gram of DNA can theoretically store more than 200 petabytes of data. In molecular fluidic computers, DNA's ability to pair with complementary strands makes it an excellent medium for both storing information and performing computations. For beginners, it's useful to think of DNA as nature's most compact and efficient storage system, capable of holding incredible amounts of data in a tiny space, all while being robust enough to last thousands of years. One of the key advantages of DNA is its ability to undergo sequence-specific reactions. DNA can recognise and bind to a complementary sequence with high precision. This ability can be harnessed to create logic gates at the molecular level. For example, when one strand of DNA binds to its complementary sequence, it can displace another strand, triggering a reaction. This is similar to how a logic gate works in traditional computing, where an electrical signal causes a change in the circuit. For those new to this concept, think of DNA as a tiny, programmable molecule that can be directed to perform specific tasks based on its sequence. DNA has a natural ability to self-assemble, meaning complementary strands of DNA will automatically find each other and bind together. This property is critical for molecular fluidic computers because it ensures that the right molecules meet and react. Additionally, DNA structure allows for built-in error correction. If two strands of DNA do not match perfectly, they are less stable and are more likely to be corrected or removed. This self-correcting ability makes DNA a reliable medium for molecular computations, ensuring that mistakes are minimised in complex systems. For beginners, it's helpful to think of DNA as a puzzle where the pieces snap together naturally and incorrect matches are rejected. One of the major advantages of DNA-based molecular computation is its ability to perform many reactions at once, known as parallel computation. In a traditional computer, tasks are often completed one at a time, sequentially. But in a molecular system, millions of molecules can react simultaneously. This parallelism allows molecular fluidic computers to process vast amounts of information more quickly than traditional computers in certain contexts. Beginners can imagine it like a large group of people all solving different parts of the same puzzle at the same time. This speeds up the process compared to one person solving it alone. DNA sequences can be programmed to carry out specific chemical reactions. By designing DNA strands to interact in predictable ways, we can control when and how reactions occur within a molecular fluidic system. For example, a DNA strand could be designed to bind to a target molecule only when certain conditions are met, such as the presence of a specific protein or environmental factor. This capability makes DNA an incredibly powerful tool for building programmable molecular systems that can perform precise tasks, such as detecting a virus in the blood and triggering a response. Understanding this for beginners is like programming a computer to run certain programs only when certain conditions are met, but instead of code, you use DNA sequences. While molecular fluidic computers offer many exciting possibilities, they face several challenges. One of the biggest hurdles is controlling the timing and location of chemical reactions. Molecules in a fluid environment can diffuse unpredictably, making it hard to ensure that reactions happen exactly where and when they should. Another challenge is scalability. While molecular fluidic computers can perform many reactions at once, scaling them to handle complex computations at the level of modern computers is difficult due to noise and error rates. These challenges must be overcome for molecular fluidic computers to reach their full potential. One of the biggest challenges in molecular fluidic systems is controlling chemical reactions with precision. In a fluid environment, molecules are constantly moving, and this makes it difficult to ensure that reactions happen at the right place and time. To address this, scientists are developing microfluidic devices that can channel and control fluid flow, ensuring that molecules meet under controlled conditions. This is similar to managing traffic in a city. Without proper signals and roads, cars would crash or get lost. Microfluidic channels help guide the molecules, preventing unwanted reactions and ensuring accuracy. Although molecular fluidic systems can process many reactions at once, parallelism, scaling them up to handle complex computations, poses significant challenges. As the system grows in complexity, it becomes more difficult to control each reaction, and errors can accumulate. Researchers are exploring hierarchical designs, where computations are broken into smaller, manageable sections that interact with each other. This approach is similar to how modern computer processors use multiple cores to handle different parts of a computation simultaneously. The goal is to make molecular fluidic computers more powerful and scalable. Molecular reactions are slower than the electronic signals used in traditional silicon-based computers. While molecular systems have advantages like parallelism and the ability to operate in biological environments, their speed is a challenge. Enzymes or other catalysts can be used to speed up these reactions, but they still operate on a much slower timescale than electrical circuits. For beginners, it's important to understand that while molecular fluidic computers may not be as fast as traditional computers, they have unique advantages that make them valuable for specific applications, especially in biological systems. Molecular fluidic systems offer the potential for energy-efficient computing. Chemical reactions, particularly those catalysed by enzymes, often require less energy than traditional electronic circuits. Think of how your body performs countless chemical reactions using very little energy compared to what's needed to power a smartphone or computer. This energy efficiency could make molecular fluidic computers ideal for applications where energy is limited, such as in wearable devices or implantable medical systems. For beginners, understanding this concept is key to appreciating why molecular fluidic computers could be important for the future of low-power computing. Molecular fluidic computers could revolutionise many fields. They could be used for in-body diagnostics, providing real-time health monitoring at the molecular level. They could also be used for environmental sensing, detecting pollutants in the air or water with incredible sensitivity. These applications are exciting because they go beyond what traditional computers can do. Traditional computers struggle in environments like inside living cells or extreme conditions like space. Molecular fluidic computers, however, are designed for these environments, offering entirely new possibilities for computing and sensing. One of the most exciting potential uses of molecular fluidic computers is in biomedicine. These systems could be placed inside the human body to monitor health at the molecular level, detecting disease biomarkers like proteins, DNA mutations, or metabolic changes in real time. For example, imagine a molecular fluidic system embedded in your bloodstream, continuously analysing blood chemistry to detect early signs of illness, long before symptoms appear. Such a system could even trigger a response, like releasing medication or alerting a doctor. This represents a huge step toward personalised medicine, where treatments are tailored specifically to an individual's biological data. For beginners, it's helpful to think of these molecular fluidic computers as miniature doctors inside your body, constantly checking for problems and providing treatment exactly when and where it's needed. In synthetic biology, scientists design and construct new biological systems or reprogram existing ones for useful purposes. Molecular fluidic computers could play a significant role in this field by enabling cells to process information and make decisions based on their environment. For example, a molecular fluidic system could be programmed to detect certain molecules inside a cell and trigger a response, such as producing a specific protein. This ability to control biological processes with such precision could lead to innovations in areas like drug production, biofuel generation, or environmental cleanup. For those new to this idea, imagine cells behaving like computers, making decisions based on molecular inputs, much like a thermostat turning on the heat when the temperature drops below a set point. Molecular fluidic systems can also be used to create highly sensitive environmental sensors. These sensors could detect trace amounts of harmful substances in the air, water or soil, chemicals that are often too small for traditional sensors to detect. Imagine a molecular fluidic sensor that could detect pollutants in the air in real time, alerting authorities to dangerous chemical leaks or contamination events long before they become a serious threat. This kind of early detection could prevent environmental disasters and protect public health. For beginners, it's helpful to think of these molecular systems as highly specialised sniffers that can detect chemical changes in the environment with incredible precision. Molecular fluidic systems offer exciting possibilities for drug delivery. Imagine a system that could release medications only when specific conditions are met inside the body, such as elevated glucose levels in a person with diabetes. This kind of precision medicine could ensure that drugs are delivered only when and where they are needed, reducing side effects and improving patient outcomes. Such systems could revolutionise how we treat chronic diseases, making treatments more efficient and less invasive. For beginners, think of this as having a molecular, smart system inside your body that releases the exact amount of medicine needed at the perfect time, without requiring injections or pills. One of the mechanisms that molecular fluidic computers rely on is DNA strand displacement. This is a process where one strand of DNA binds to a complementary sequence, displacing another strand in the process. This reaction can be used to perform logic operations like AND, OR, or NOT, similar to how traditional computers use electrical signals. The beauty of DNA strand displacement is its precision. It's like a lock and key. Only the correct DNA sequences will fit together, triggering the next step in the computation. For beginners, this concept might be compared to how a specific password unlocks your computer. Only the right combination of molecules can trigger the correct reaction, ensuring accurate processing at the molecular level. Enzymes are proteins that speed up chemical reactions, and in molecular fluidic computers, they can act as logic gates. For example, an enzyme might only catalyse a reaction if two specific molecules are present, functioning like an AND gate. This type of computation could take place inside living organisms, allowing cells to process information and make decisions in real time. It's a bit like giving cells a brain that can react to environmental changes and carry out complex tasks, such as producing hormones or fighting infections. For beginners, enzymes can be thought of as tiny workers that speed up production lines, ensuring that tasks, reactions, happen quickly and at the right time. CRISPR, known primarily for its gene editing abilities, can also be used as a molecular processor. CRISPR proteins can be programmed to recognise and target specific DNA or RNA sequences, functioning as molecular logic gates. This means CRISPR could perform complex molecular computations inside cells, such as detecting harmful genetic mutations or even regulating gene expression in response to specific inputs. For beginners, think of CRISPR as a molecular search and edit tool, not only capable of cutting and pasting DNA, but also computing, determining when and where these edits should happen based on molecular signals. DNA origami refers to the process of folding long strands of DNA into specific shapes, which can then be used to organise and control molecular reactions. These nanostructures provide a way to arrange molecules in precise patterns, enabling complex computations to occur at the molecular level. This is similar to how you might fold a piece of paper into a particular shape to create a structure, but at a scale so small that the paper is a strand of DNA and the shape is a scaffold for molecular circuits. These tiny structures could be used to build molecular machines that operate inside living cells, performing tasks like drug delivery or even assembling new materials. For beginners, imagine building a tiny robot out of DNA that could move through your body, assembling proteins or delivering medicine exactly where it's needed. A molecular Turing machine is a theoretical model where molecules act as the tape and head of a Turing machine, the fundamental model of computation used by traditional computers. In this system, molecules carry out operations based on a set of rules, just like a Turing machine processes instructions on a tape. Such a machine could, in theory, solve any computational problem, just like a regular computer, but at the molecular level. It's a powerful concept that could push the boundaries of computation into entirely new territory, such as operating inside biological systems or at nanoscale levels. For beginners, think of a Turing machine as a very simple but universal computer, and imagine it being rebuilt entirely from molecules that interact and compute based on chemical reactions. As molecular fluidic systems shrink to the nanoscale, the rules of quantum mechanics begin to take over. Quantum effects, such as superposition, where a molecule can exist in multiple states simultaneously, and entanglement, where particles are interconnected across distances, could be leveraged to make molecular fluidic computers even more powerful. Quantum phenomena could allow molecular systems to perform computations that are impossible for classical computers, such as solving problems involving enormous amounts of data. For example, quantum molecular computers might be able to simulate complex chemical reactions or biological processes with unprecedented accuracy. For beginners, it's useful to think of quantum mechanics as a set of weird rules that only apply at extremely small scales. But these rules might give molecular fluidic computers capabilities far beyond what today's electronics can achieve. Molecular fluidic computers represent a new frontier in the world of computing. By harnessing the natural properties of molecules, we can build systems that operate in environments where traditional electronics fail, such as inside living cells or extreme conditions like space. This technology has the potential to revolutionise fields like medicine, environmental protection, and even artificial intelligence. Imagine personalised healthcare, where molecular computers inside your body monitor your health in real time, detecting problems and delivering treatments before you even know you're sick. As researchers continue to refine the ability to control molecular reactions, improve scalability, and integrate quantum effects, molecular fluidic computers will become more practical and widespread. The future of computing may be biological, and molecular fluidics could be the key to unlocking that future.